This is a short video on how to obtain organelles from a tissue. For example, you might have a bit of liver and you want to get the organelles out of the liver cells. So what do? The first thing that you do is you homogenize the tissue. That really means you liquidize it. You put it into a blender and spin it. And as the blades go through the tissue, they will open up the cells. But the tissue should be placed in a solution which is ice cold, isotonic, and it is buffered. Why should it be ice cold? It should be ice cold to stop chemical reactions so that all of the organelles remain intact. Secondly, it should be isotonic so that there is no osmotic damage. If the solution had a really high water potential, the water would move into the organelles and it could cause them to burst. And the last thing is that solution should be buffered so that the enzymes inside those organelles will not be denatured. So after all of the organelles have been released into that ice cold isotonic buffered solution, you then have to separate the organelles out, and you do that using centrifugation. But before you would centrifuge this solution, you would of course filter it to get out any large bits and pieces of cell debris. So you put this solution into centrifuge tubes and then you put them into the centrifuge and you spin at high speeds. Now, whenever you spin this solution at high speeds, you'll end up with a sediment at the bottom, which is just called the sediment, and the solution at the top is called the supernatant. Now, first of all, when carrying out centrifugation, make sure that every tube is counterbalanced with a tube which has the same volume of solution. And that means when the centrifuge is turning round and round and round, it will spin evenly. Now, what you do is you spin first of all at a lowish speed, and whenever you do that, the largest organelles will sediment out. So the largest organelle there would be the nuclei. So that means if you pour off the supernatant, what you're left with is a sample of nuclei. So you have separated out the nuclei. So the supernatant then doesn't contain nuclei, and you spin it at a faster speed, just the supernatant. And when you do that, in the sediment you will have organelles then which are smaller than the nuclei, for example mitochondria. So again, that supernatant could be poured into another tube, and there you have mitochondria in the sediment that could be used in your experiments. So that supernatant, which does not contain mitochondria or nuclei, is then spun at a very fast speed. And whenever it's spun at a very fast speed, the smallest organelles come out into the sediment, and that would be things like ribosomes. Now, it's very important to remember you do not use the fastest speed first, because if you use this really fast speed here at the beginning, your sediment would contain nuclei, mitochondria, and ribosomes. All organelles would end up in the sediment. So you start with a low speed, and then as you speed up, different organelles end up inside that sediment. I then just want to mention how you stain cells. So the purpose of staining cells, say I like you to say, it makes structures more visible. And two stains that you definitely need to know are iodine. Iodine is very good at staining plant cells and making the structures more visible. And methylene blue is very good at staining animal cells. It particularly makes the nuclei of those animal cells more visible. Obviously, you also need to know your ordinary practical on how to make a slide of onion cells or cheek cells. So you make sure that the tissue is flat. Remember that you add the stain. You lower a cover slip with a mounted needle to make sure that there are no air bubbles. And then when you set it onto the stage of the microscope, you start with the low power objective lens. Make sure that you know that very easy practical.